Hi everybody. In this video lecture we will be talking about Cyanophycean. This is the second part of this video lecture about Cyanophycean. In our first video lecture we talked about the general features of this group. Here we will be discussing about the ultrastructure of the vegetative cells of Cyanophycean alga. But we will also discuss about the Thallus organization of this group, plant body types that ranges from unicellular to trichomatous to filamentous forms. There are some falsely branched filaments as well as some truly branched filaments, but heterotrichous forms are altogether absent in the group. Now let us come to the point. So we wanted to discuss the Cyanophycean alga. The general features have already been discussed. The ultrastructure of the Cyanophycean cell has to be discussed now, but before that we are going to discuss the Thallus organization. So some of the members of Cyanophycean are the unicellular like this. When they divide, they take a shape like this when a binary fusion is going on. This is about Gliocapsa we are talking about. This is the structure of Gliocapsa, the unicellular member of Cyanophycean. There is another member, Crucoccus, but the structure has not been drawn here. These are the unicellular members. Now there are some trichomes. There are two types of filamentous bodies called trichomes and filaments. These two things are different, but literally it seems similar that trichome is also a kind of a thread-like structure, chain of cells that are arranged one after another, giving more or less filamentous body, but that cannot be technically called filament because it lacks the mucilaginous sheath around it. If this mucilaginous sheath is present, as shown here in the nostoc, it will be called filament. So difference between the trichome and the filament is the absence of the mucilage sheath around the chain of cells or thread-like structure. So this is represented by the trichomatous form is represented by Anabina, which is being shown here. Anabina, a very important alga with heterocysts. These are heterocysts, specialized cells for nitrogen fixation. This is a reserve food, asexual reproductive unit called echinate, sometimes also treated as a vegetative reproductive unit, echinate. And these are the vegetative cells of the alga. So this is the trichome of Anabina, and this is the unbranched trichomatous structure. There are some unbranched filamentous structure like the oscillatoria. This one is oscillatoria. Several disc-like cells are arranged one above another. They are stacked one above another, and from one side they look like this. These are the disc-like cells. Actually, they are disc-like like this, and one above another when they are arranged. And when you look from one side, they look like this. There are some biconcave cells. These are called separation discs or necridia. So the cells in between these two separation discs, they get separated from one another. They get separated as a unit. That is called hormogonia, and they act as a reproductive structure. Basically, these are also present in a shade-like structure. So this is also a filament, and this is nostoc filament. A very important alga in the rice fields. They help in nitrogen fixation because they also contain these heterocystous cells, intercalary as well as terminal heterocystous cells. Nostoc balls covered in a mucilaginous structure like this kind of filament. There several such filaments are present inside in a ball-like structure they form. You can find them in rice fields. So there are some other filamentous alga. This is called Gliotrichia, and this one is Ribularia. The speciality of this alga is that they contain terminal heterocystous cells. This is the heterocyst. This one is also heterocyst. But the difference between them is that in Gliotrichia you will find an echinate cell just after the heterocyst, which will be absent in case of Ribularia. Otherwise, both are very much similar. But there is another structure, the sheath-like structure is present in case of Gliotrichia just after the heterocyst, covering a basal portion of the filament. And then let us come to the branched filamentous bodies or structure or organisms, species of Cyanophyceae. This is Cytonema. It exhibits false branching. As you can see, the two filaments they have entered, two trichomes in fact, they have entered in a common sheath. This is the sheath, common sheath, within which they have come together. But and then they pierce it here and come out from this sheath-like body, and as if that it appears they are branched here, but this has not come out from cell proper. So this is not a true sense of branching, and in fact two trichomes have entered in a single common sheath, and they pierce the sheath and come out like a branched structure, but the branching is not from cell proper. The true kind of branching is seen in case of Stegonema, which has been shown here. In Stegonema, you can see that this branch has come out from a cell proper. In fact, they are multiseriate cell-like structure, and they are also covered with a sheath-like body, which is which up to some extent is not covered. Also, this part is free, but this part has been covered with the mucilaginous sheath. So they are also filamentous structures. They are truly branched filamentous structures of Cyanophyceae. Now let us come to the ultrastructure of the vegetative cells. We are talking about the ultrastructure of this kind of vegetative cells, these cells, or this cell. Here a drawing has been done with respect to the apical cell, say this apical cell of Oscillatoria. This particular apical cell has been drawn to show the ultrastructure. But in some textbooks you might find the intercalary cells being drawn, or unicellular member may be drawn to show the structures, different structures of the cell. Doesn't matter. The main feature of this Cyanophycean cell is that the cell wall is cell envelope, you can say, is gram-negative type. This is the plasma membrane. Outside the plasma membrane, there are several layers. In some book, these layers have been divided into, in previous literatures, in fact, they were divided as L1. This inner layer is called L1. This layer is called L2. This empty layer is called L3, and this outside sheath is called L4, like this. But recently, it has been found that this L1 and L3, you may call them periplasmic space or periplasm, just like the gram-negative bacteria. And this orange color L2 layer is basically peptidoglycan. This is also similar to gram-negative bacteria, but it is very much thicker, unlike gram-negative bacteria, which are merely 6 to 8 nanometer thick. Here it is about 20 to 40 nanometer thick, and then similar to gram-positive peptidoglycan. But the difference with the gram-positive is this, that the periplasmic space is distinct, and also there is an outer membrane very much similar to gram-negative bacteria. So this is outer membrane. Some of the cells, some of the species might contain a sheath-like. This is a sheath-like body outside the outer membrane, and the organism that contains sheath, they also have another layer of outside this outer membrane. This is called S layer. This is a proteinaceous layer called S layer. After that, the oscillin fibrous layer. This sheath-like structure contains a proteinaceous fiber called oscillin fibers, and they help in the gliding movement of the organism. But these two structures, the S layer and the sheath or the oscillin fibers, are not present in all members. Anyway, let us see the structure of the protoplasm. The protoplasm can be divided into peripheral protoplasm, and that can be called ectoplasm, and the inner protoplasm, the central protoplasm, that can be called endoplasm. So the outer peripheral protoplasm has several lamellated membranous structure. They may be layered or they may be scattered, but if they are layered, even there must be some cytoplasm in between them, and this structure is called thylakoid. So the thylakoid is not organized in the chloroplast form. There is no membrane bound structure like that, and the thylakoids contain several globular structure on their surface. These are basically phycobilisomes. These phycobilisomes, they contain protein pigment structures. The pigments that they contain, the phycobili proteins that they contain are the C-phycoerythrin, C-phycocyanin, L-phycocyanin, and phycoerythrocyanin. These many pigments are present here, and also the membrane. This structure is basically thylakoid membrane, is also similar to the plasma membrane. These two structures are very much similar because they are made of phospholipids like this. So they embed chlorophyll molecules inside them. This is suppose chlorophyll molecule. Chlorophyll A is present rather than bacterial chlorophyll. Now let us talk about these structures. This one is a nitrogen reserve called Cyanophycean granules. They are nitrogen reserves, basically made of mainly of aspartic acid and arginine in 1:1 ratio. And these polyhedral bodies, these are polyhedral bodies. Also called carboxysomes, because they contain the enzyme rubisco, that is the enzyme for carbon dioxide fixation. Now let us talk about this structure. This is polyphosphate granule or polytune granule, poly-P bodies, also called polytune granule. They contain a polymer of orthophosphate. This is the nucleic acid, that is the DNA. It is not bound by any nuclear membrane, so if the DNA, the chromosome is naked, chromosome is naked just like bacterial system. This structure is called pseudovacuole, and they are made of several vesicles stacked one above another. These are called pseudovacuole. In fact, the Cyanophycean cell does not contain vacuoles like the plant cells or the cells of other alga, and instead they contain pseudovacuole like this that are made up of proteinaceous membranes. Two types of proteins are present here in these vesicles. These are GVP-A and GVP-C. They make the membrane-like structure of these vesicles, and inside that there are carbon dioxide gas and several other gas that helps in buoyancy of the cell. Because of this, the filament of Cyanophycean may float at different depths in the water bodies. These are the ribosomes. The 70S type ribosomes are present, and these are alpha granules, we may also call glycogen bodies, glycogen reserve foods, also called alpha granules. These are carbon reserves. So basically this is the ultrastructure of the Cyanophycean cell. We may go to even further details, but I think for undergraduate level this is more than enough, and hope it will help you to some extent.